Thank you. Yeah, so I'm Alexander Hübner. I'm a postdoc in the group of Christina Warner. And as the last talk before lunch, I want to introduce to another human community that actually has an almost fiber-free diet that they live on. By studying the evidence of animal milk proteins in ancient dental calculus, our lab could show a few years ago that actually nomadic pastoralism arrived in the Eurasian steppe about 5,000 years ago, and it's still a very common way of subsistence, particularly in Mongolia today. And naturally, in mammalians, the milk is the primary source of nutrients for the infants. It consists out of milk oligosaccharides, fats, and proteins. And one of the milk sugars that is in relatively high abundance is actually the disaccharide lactose. And to utilize lactose as a nutrient, you actually have to break down the glycosidic bond with an enzyme, and that many of you know, it's lactase. And typically, lactase is expressed in the small intestine of infants, and then gradually the expression is decreasing with increasing age. This happens unless there's a few known genetic variants in the human host genome that actually allow the persistent expression of lactase in adulthood, and then you can do what the Danish government recommends to you, drink half a liter of milk a day without getting troubles. If you don't have this lactase in the small intestine, then the lactose is passed on into the colon, where the microbes start breaking down the lactose. And during this microbial fermentation of lactose, gas is produced that then causes bloating, nausea, and really impacts the lifestyle of the individual suffering from that. Despite a long-standing tradition of pastoralists in Mongolia never really reached any high frequencies of any of these variants that allow lactase persistence, and at most they were at 5% of the population. Instead, what they are doing is they apply a culture technique that microbial fermentation to actually break down the lactose outside of the human body before digesting it. And over the years and centuries, millennia, they developed a large variety of preparing different fermented dairy products, such as tarak, esky, or a very particular specialty, fermented horse milk called irak. And to kind of emphasize how important these microbial fermentation is for them, is that the starter cultures that are used for it are then even passed down as heirlooms to the daughters when they leave the household and move out from their parents into the in-laws as a typical gift that is handed on to by the parents. And if you look at the starting cultures, they are rich in lactic acid bacteria and yeast. And the individuals, the herders, are consuming quite a lot of these dairy products. So we were asking, does it actually have also, next to the effect of having no lactose in there, have a probiotic effect on the gut health? And to answer this question, or try to answer this question, we actually went to Mongolia with local colleagues and sampled their 50 different pastoralists in 3 different regions during the summer month, where they almost exclusively, like 98% or 99% of their diet is actually dairy-based, so no fiber at all. And compared them to 50 individuals from the capital city, Ulaanbaatar, that very much have a lifestyle like we all here today, and they go to the supermarket and consume like an industrialized diet. We started extensive sampling and collected stool and saliva samples from the herders. We also took a lot of frozen samples from the dairy products and swapped the animals, the household appliances, filtered the air, and collected a lot of samples to really understand where the bacteria are coming from. And to answer the first question, which was really focusing as a start of the whole discussion on the lactic acid bacteria, we wanted to know which lactic acid bacteria are actually present in the dairy products and which are present in the human gut. And for this, we cultured the dairy products, the bacteria in the dairy products, and then sequenced the isolates and the Oxford Nanopore platform, while the stool samples were collected in FDK cards, shown here in the bottom, were directly extracted without cultivation and shotgun sequenced on the Illumina platform. We then took the Illumina sequencing platform and sequencing data and actually reconstructed max from them. And from the over 8,200 max that we could reconstruct, about 230 were actually lactic acid bacteria genomes. And as you see here on the three panels on the left, these are the herder communities. They have a slightly higher prevalence of lactic acid bacteria in the gut than actually the urban population. Next, we want to compare what we actually found in the isolates that we obtained from the dairy products. And what we could see here is, while there are some species that are exclusively found either in the gut or some in the only in dairy products, there are some like Lactobacillus lactis here or Enterococcus faecium, which are found and shared across the two different sample environments. And it's nothing new that lactic acid bacteria are found in human guts. It's well studied. However, there's a difference between different species on how prevalent they are. So here you see a comparison on the prevalence of certain lactic acid species compared to the prevalence in a comparative panel of worldwide population split by continent. And you see that a lot of species like Lactococcus lactis or Streptococcus thermophilus that are usually used as in commercial starting cultures, like the ones from Christian Hansen, who sponsored the auditorium here, are very present. On the other hand, some species are actually almost exclusively found in Mongolia, but absent in other regions. And one of them is Lactobacillus helveticus. So then we were particularly interested in this one and reconstructed, took all of the genomes, reconstructed, and did a phylogeny on them. And what we see is all these genomes here in darker blue that come from Mongolian gut microbiome form a monophyletic clade that falls outside of the known variation that was found in literature before. But interestingly, the only species we so far have isolated from the dairy products actually falls completely outside of the variety and is not part of the clades, suggesting that there's some divergence between them. To conclude and summarize my talk, we learned today that lactic acid bacteria are highly prevalent in the Mongolian gut microbiome, and there is a significant overlap between the lactic acid bacteria found in the dairy products and in the human gut microbiome, suggesting that there is definitely some sort of effect that we, with our preliminary results, couldn't yet really investigate further. As a next step, what we really want to do is optimize, first of all, the culturing techniques to obtain a more diverse variety of lactic acid bacteria, because we had a lot of very similar species around. Because one of the parts of this project is also to return back this information on which lactic acid bacteria are found in Mongolia, to return it back then to the public domain, because many of the herders are actually switching when their starting cultures fail to commercial starting cultures like the ones sold by Christian Hansen, and not go back to their indigenous ones, and so we want to return back that. The next step also include, we want to actually understand, are these lactic acid bacteria found in the gut microbiomes, are they adapted to it, or are they just transient ones? So more or less, if I drink 3 liters of milk every day, fermented milk every day, do I just see the effect of having the bacteria there, or are really these species adapted? And we hope to look further into this. And finally, we want to know whether or not the animals and the household appliances are actually also reservoir for lactic acid bacteria strains and how stable their starting cultures are in the long run. And with this, I would like to thank my boss, Christina Warner, and our whole team, all the funding bodies, and particularly the herders that gave all this insight and were so willingly to share samples with us. And you for your attention.
Thank you very much, Alex. We have time for one question before we go for lunch. Let's see if there is anyone. We have 2 questions. Itzig, you have already asked questions today, so let's go. We have for lunchtime. Yes, it's getting there. Come on. Go. There we go.
Is there a reason that you cultured the microbes first, rather than just sequencing them directly, like you do with the stool samples?
So we have a shot, like a few of the samples we sequenced directly. However, it turned out that it's not as simple to extract DNA out of these complicated or complex samples, because they are usually fat-rich or they have some properties where you have to adapt very much on every of these dairy product spaces the extraction protocol. And so in the end we said, okay, we give as a part of the pilot that gave us the ERC funding, we did just batch culturing of everything. But at the moment, we are going back to systematically do exactly what you suggest, do a shotgun screening, know roughly where you are, and then optimize the protocol, the isolation culturing protocols based on that.
Thank you. Yeah, thank you very much, Alex. I'm going to take over briefly.